So welcome back from the break. Hopefully you took time to step away for a few minutes. Let's take a look at the concept check question and keep moving onwards. So compare and contrast the two body plans of the Medusa and the polyp of Cnidarians. So let's go and take a look at the diagram again. We can go back to that. So here are the two body plans. So both of them have a two-layer body plan, the epidermis on the outside derived from ectoderm, and the lining of the gastrovascular cavity, the gastrodermis, derived from endoderm. They both have a single opening into the gastrovascular cavity, the mouth anus opening. That defines the two sides of the organism. The oral side is where the mouth is, and the aboral is the opposite side. In the case of the polyp, of course, you have an attachment point to the substratum via the aboral side of the polyp, and it creates a sessile form of the cnidarian. In the medusal form, the aboral side is the top of the bell, the oral side is the mouth entering into the gastrovascular cavity. It always points downwards in terms of the orientation of the medusa. Remember, both of them are surrounded by tentacles. On the oral side, those tentacles contain the cnidocytes with the nematocysts that provide the stinging apparatus for disabling the prey. So there are the two body plans of the cnidarians. So let's go onwards and take a look at the next group of animals that we need to pay attention to. So now we move from radial symmetry to bilateral symmetry. And remember, bilateral symmetry means that we have three axes. So just fire up the pen for a minute. So the three axes are anterior to posterior, and that's the axis that defines the bilateral symmetry. And then we have dorsal ventral. And left and right. And remember, it's the left-right symmetry that is also associated with the bilateral symmetry. So mirror image around and post axis. So that's where the bilateral symmetry comes in. And we have three major clades that we need to address here: the lophotrochozoans, the ecdysozoans, and the deuterostomia. So we'll begin by looking at the lophotrochozoans, then we'll address the ecdysozoans, and then we'll do the deuterostomia. And we are going to get bogged down in the ecdysozoans. And the reason why is because that's where we encounter the arthropods, and very specifically, we encounter the insects. So that is going to be a long presentation. And I may actually take a break in the middle of the arthropods as I think about it. We'll see. And separate out the history of the arthropods from a look at the crustaceans and the insects. We'll see when we get there. Anyway, let's begin with the lophotrochozoans. So what groups these together? Well, part of it is, of course, DNA sequence analysis, which establishes the validity of the clade. But also, in the case of the lophophore portion of the clade, it's the presence of the lophophore feeding organ. And in the case of the trochozoan side of the clade, it's the presence of the trochophore larval stage. The major clades that we're going to look at are the flatworms, platyhelminthes, the rotifers and acanthocephalans. This is the new clade, and I've forgotten the name of it now, but we'll get to it. Then we have the lophophore clades, the ectoprocs and the brachiopods. The mollusks, which is a reasonably large clade, but we don't dig terribly deeply into it. We will spend a little bit of time with it. And then the annelids. So let's begin with the flatworms, otherwise known as platyhelminthes. These are triploblastic in their development. They are also acoelomate, and the current understanding is that the acoelomate body plan is a derived trait. As I pointed out in our coverage of chapter 32, we have had to rethink what the original animal body plan might have been. And the current thinking is that it was likely bilateral, segmented, and had a coelom, a true body cavity. And that in fact, the hemocoel or pseudocoelomate body plan and the acoelomate body plan, or solid body plan or compact body plan, are actually derived traits, not ancestral traits. And so this is the reason why we no longer consider the presence or absence of a body cavity to be a way of referencing the phylogenetic history of the animal kingdom, because it simply is not a valid way of looking at it anymore. And there's some DNA sequence evidence that is now starting to support this view. So it's not just what we see in the fossil record. DNA sequence analysis is actually starting now to support the view that the alternative body plans to the coelomate body plan are in fact derived body plans, that flatworms are derived from ancestral bilateral segmented coelomate ancestors, and dispensed with segmentation and dispensed with the body cavity completely as a adaptation to the lifestyle and the habitats that they now occupy. They are flattened dorsoventrally and also have a gastrovascular cavity, which means that none of the cells within their body are more than 2 to 3 cells away from an outside surface, because the gastrovascular cavity, remember, opens out into the outside. And so then no cells are more than 2 to 3 cells away from fluid. And that means that gas exchange and waste disposal can take place by diffusion across the surface of the animal. And when you look at the nature of the gastrovascular cavity inside the turbolaria, which are the ones that actually have the gastrovascular cavity, a substantial gastrovascular cavity, it is highly branched, and then it has a lot of little sub-branches, creating a large surface area to support diffusion of nutrients out of the gastrovascular cavity into the body of the worm. But they do have what might be referred to as some primitive excretory cells called protonephridia. And so nephridia refers to secretory cells that ultimately will be found in or precursors to secretory cells that will ultimately be found in kidneys and secretory organs like that. Proto means ancestral to, and so these are primitive secretory cells, but their job is to regulate the movement of water into and out of the organism by moving solutes in and out of the organism. So they are all about osmoregulation, not about waste disposal. In terms of the phylum, there are 4 classes that we recognize. The first is the turbolarians. These are the widely recognized, otherwise known as planarians. They are found in aquatic environments and also in terrestrial environments where there is a substantial amount of water. In fact, in Great Britain, there is an issue with a turbularian that was accidentally introduced into the country from New Zealand, and it is a predator that preys on earthworms. And it is in the process of migrating from Scotland, where it was first introduced, probably on potted plants, down through northern England. It's down into the Midlands now, and it is changing the shape of agriculture in some locations, because in destroying the earthworms, it's destroying a major source of aeration of the soil and churning of the soil. We'll talk a little bit more about that when we get to the annelids. And that means that the water that would normally drain into the soil is no longer able to do so, and it's changing the nature of agricultural land in some locations, all because of one of these earthworms, and again, flatworms. And again, there is a video posted in the resource section that talks about the flatworms as some of the first predators that may have emerged in the history of life. And it actually addresses these flatworms that are preying on the earthworms. And you actually watch a flatworm hunt down and capture an earthworm as part of the video, again, from the shape of life. So it's well worth taking a look at that. So this is the typical body plan of what is called Dugesia. So this is Dugesia. Oops. I think that will have got all of it. Let's try this again. So if we were in lab on campus, we would have a little metal tray with probably about 30 or 40 Dugesia swimming around in it, and you could see the shape of the animal, and it has a body plan like this. So you can see the gastrovascular cavity. You can see what I mean by it being very, very highly branched to penetrate into the body of the animal. Excuse me. It has a single layer of muscles that run longitudinally that allow it to undulate if it's swimming in the water column, or it can cruise on cilia on the underside of the animal across the surface, and it can actually secrete a mucus material that allows it to glide across the surface. In terms of coordinating activity, there is a pair of nerve cords. There are a cluster of nerves at the anterior end that form a structure called a ganglion. This is the functional equivalent of a brain. It coordinates the activity. It sends nerve signals to coordinate the muscle contractions. Notice that the nerve cords are described as being ventral. This is a consistent theme that you're going to see throughout the invertebrates. The nerve cord is always ventral. That will change when we get to the chordates, as you'll see. In the center of the anterior end and representing the beginnings of cephalization, remember the clustering of sensory apparatus at the anterior end is something that we see with bilateral animals, and we refer to that as cephalization. We have a pair of light sensing organs called eye spots, and then out on the edges of the triangular region of the quote unquote head, we find auricles. And this senses make sure we get rid of all of that. And the advantage of having auricles on either side of the head is that it gives you a stereoscopic view. It allows you to determine the direction from which the chemicals are coming, because you can, if it's coming from straight ahead, both auricles will sense it equally. If it's coming from one side, the auricle on that side senses it more intensely than the auricle on the other side, which allows you to get directional information. And so when a planarian is on the hunt, like that flatworm that chases down earthworms, you can actually see it moving its head backwards and forwards and sensing the chemicals that have been left behind by the earthworm. And this provides the flatworm with the information that it needs to zero in on the location of the earthworm. These all feed by secreting digestive enzymes into the prey material. So digestion takes place externally, and then the digested material, a sort of a soupy material, is then sucked up via the pharynx into the gastrovascular cavity to be distributed throughout the body to provide nutrition. Yeah, one other thing. There is another video that is posted in the resources section for this chapter that highlights, again, from The Shape of Life, a very unusual behavior of flatworms that live on the Great Barrier Reef and elsewhere. It was first identified on the Great Barrier Reef. This is called penis fencing. Yes, you did hear me correctly. Penis fencing. Because flatworms are hermaphrodites, both males and females, and particularly in the marine ones, they exchange sperm. But because it takes resources and time and effort to carry the burden of being the quote unquote mother, the worms actually fight to see who gets the sperm and who gets to walk away. And so they grow a pair of appendages that look like enlarged daggers. These are actually penises. There are a pair of them on the underside of the worm, and then they rear up and they literally wrestle and fight and try and jab the other worm with these penis daggers. The first one to jab releases sperm into the body of its victim. The sperm immediately gets absorbed, and you can actually see on the video the sperm being absorbed into the body of the worm that got jabbed. And that sperm then fertilizes the eggs within the body of that worm, and it now has to function as the mother and carry the burden of being the mother to these young worms until they hatch out. The other worm goes off to live and fight another day, having successfully jabbed its opponent. So it's an interesting video to look at. Yes, penis fencing, a very unusual behavior, not something that you would have expected from something like a simple little flatworm. The next two classes are the monogeneans and the trematodes. These are parasites. So monogeneans are parasites of fish in both marine and freshwater environments. If you've ever seen a video of a reef ecosystem, a documentary looking at a reef ecosystem, scattered along the reef are special locations called feeder stations, and there little fish called cleaner wrasse, little crustaceans and so forth will gather around the larger fish that come to the cleaner stations, and those large fish will simply hover in the water column, open their mouths, and let these little shrimp and the little fish go to work, cleaning off all the parasites and detritus that's on the surface of the fish. And they don't try and eat them. They understand that this is neutral ground, that this is a beneficial relationship for both, and no harm should be done in either direction. And one of the reasons why The large fissure there is to get these monogeneins taken off their outer skin because they can be quite irritating. Trematodes are parasites of vertebrates, and that includes humans. So if we take a look at a typical trematode parasite that interacts with humans, the liver fluke. So this is the liver fluke. Oops. Let's erase that. Yeah. Liver. So the liver fluke is dioecious. There's male and female. The male is quite large, but he has a little groove that runs along his body that the female fits into. So this coming together of the male and the female takes place within the blood vessels of the liver in humans. The resulting zygote then develops into a ciliated larva. You can see that here, which exits or I should say the eggs exit, not the larva, the eggs, the fertilized eggs exit. So let's change that. In urine and feces. If they land in a suitable environment, which typically means water, then the zygote begins dividing and develops into the ciliated larva. This ciliated larval stage then infects the gut of the snail. And that gives inside the gut of the snail, it develops into this motile larva that exits and this is the one that then infects a human host, either by burrowing through the skin, which it's perfectly capable of doing, or entering in through a cut. That gets it into the bloodstream. It moves through the bloodstream to the liver because all the blood passes through the liver on a regular basis because the liver is a major filter for the bloodstream. And so that carries the larval stage into the liver where it matures into the adult form and the males and the females get together within the blood vessels in the liver and the cycle starts again. It's a very, very complicated life cycle. And you may ask, how does the fecal material and the urine get into the water so that it can infect humans? Well, you can see here that, that the diagram illustrates a worker working in a wet rice paddy field. And it is not unusual for human fecal matter to be used as a source of fertilizer. This is still commonly used in many countries in Southeast Asia. It is an excellent source of fertilizer and it is used as such and it is collected. And there are people men who go out at night and collect what's referred to as night soil, human fecal waste, and that is then used as a fertilizer. And in some cities in India, this has even been turned into a major way of dealing with the sheer volume of human waste that's being generated in some cities. They've actually created an ecosystem outside of the city. The human waste goes there. It's sort of a swamp, but it's a human created swamp, and the fecal material, human waste material goes in there to be processed within the swamp. The swamp is also used as a place for growing freshwater fish. Those freshwater fish are then harvested and taken to the market to be sold for human consumption. And so the cycle continues. So using human waste as a source of fertilizer is a very, very common practice. But of course, if individuals are infected with the liver fluke, using their waste, which contains the fertilized eggs, is simply going to promote the life cycle, because that gives the first fertilized eggs access to a wet environment and access to water snails, because those are endemic to large body, to even small bodies, I guess, bodies of fresh water across Southeast Asia, and so the life cycle can continue. The last group are Cestoidea, the tapeworms. These are the one group of flatworms that have no digestive tract at all. They are basically one large collection of reproductive organs with an attachment called the scolex at the anterior end of the animal. So it uses the scolex, which you can see here, to anchor itself to the inside of the digestive tract of a vertebrate, because these are all parasites of vertebrates, including our domestic pets, our domestic animals, and of course ourselves. And we can contaminate ourselves with fertilized eggs from our pets if we are not careful about how we interact with our pets, particularly if they are not being treated for worms on a regular basis. This is one of the reasons why you should regularly deworm your cat and dog. My dog is actually on a monthly deworming pill, and that makes sure that not only does she not have heartworms, which are little nematode worms, which can clog up the arteries of the heart and kill the animal, but also any gut parasite worms, like these guys, the tapeworms. How big can tapeworms get? The longest one on record was over 20 meters in length. And that was in a human gut, and it was folded up inside both the small intestine and the large intestine, and it occupied so much space that it essentially prevented the passage of food down the gut. And that's not a good thing, because the backup of material inside the gut could cause the gut to rupture, and then you can get peritonitis, which is an infection of the gut cavity, of the body cavity, that is very difficult to treat with antibiotics, and it can be lethal if it's not treated properly. There is an old wives' tale that suggests that back in the day, people used to deliberately infect themselves with a tapeworm larva in order to lose weight. I don't know whether that's correct. I've seen it written about. I've seen a lot of people say that it's not correct. I've seen people say that it is correct. I've seen people say that it's still possible to buy them. My understanding is that they are not stable outside of the body of a host for a significant length of time, besides which they've got to be grown in the digestive tract of the vertebrate in order to be able to produce the infective fertilized eggs, these proglottids that contain the infective eggs that you can see identified here. So my best guess is that this is probably just another urban myth. However, if you do get infected with a tapeworm, which you can get infected with from your pets, as I mentioned, or by undercooking meat that contains infectious material from a tapeworm, then you will lose weight, because you'll be in competition with the tapeworm, and the tapeworm will get more of benefit of your nutrition than you will. And you will be constantly hungry and constantly eating, but you will be constantly losing weight because the tapeworm will be getting the benefit of all the nutrition. You won't. The good news is that if you should be unfortunate enough to get infected, there are drugs that will kill the tapeworms. and so you don't have to suffer with them. Then we have the Cynodermata. These are the rotifers and the acanthocephalans. This is the new phylum that's been created because we now consider the rotifers and the acanthocephalans to be a single clade. So rotifers are animals that you have possibly seen. You may have seen one of these in Bio 1 looking down a microscope and seen it in pond water or something like that. These turn up quite frequently as a contaminant in some of the live specimens that we get in for various labs that we do in Bio 1. We also see them in some of the live specimens that we get for Bio 2 as well. So these are not, it's not unusual to come across these in some of the labs that we do on campus. The name rotifer means crown bearer, and that refers to this ring of cilia that surround the mouth. So the mouth is located here. Behind the mouth in the pharynx is a pair of jaws, but these jaws are grinding organs. So they go like this and force the food through as they grind it down into the stomach, which then passes on to an intestine, and then you have an anus. So they have a true digestive tract because it's got a mouth and an anus and a cylinder to connect them together. These are pseudocoelomate. They have a hemocoel derived from the blastocele. So these have the false body cavity. That's what pseudocoelom means. There is a group of rotifers called the bdelloid rotifers that reproduce by parthenogenesis, and you can see that it's referred to here. So let me put a note here. Bdelloid rotifers. So this is one subset of rotifers that do this. And so parthenogenesis refers to the ability of females to reproduce using unfertilized eggs, which obviously develop into more females. And so in this group of rotifers, there is no record of male rotifers at all. And the question has been asked, how can these continue to reproduce asexually? Because based on genetic analysis, they've been doing this for the best part of 50 million years. And that flies in the face of the idea that asexual reproduction is not the best way to reproduce because it lacks the ability to produce genetic variability, which is the hallmark of success for any species. In the absence of sexual reproduction, using just asexual reproduction, there's no real major source of genetic variability other than crossing over and random alignment of chromosomes in meiosis. And so you have a very limited source of genetic variation. And as a consequence, asexual reproduction and species that reproduce asexually should not survive for very long periods of time, like 50 million years in evolutionary terms. Simply shouldn't happen. Yet the bdelloid rotifers have been very successfully doing this for around 50 million years, as best we know. It was originally thought that this was because when you look at bdelloid rotifers, they lack any evidence of parasitism. They don't seem to be attacked by parasites. And this triggered people to take another look at organisms that reproduce sexually versus those that reproduce asexually. And immediately people started to recognize that those that were reproducing asexually were subject to reduced levels or no levels of parasitism compared to those that reproduce sexually. And so a idea arose, a hypothesis arose called the Red Queen hypothesis, or the Red Queen idea, which is based on a character from one of the Alice in Wonderland books. The Red Queen, when Alice encounters her, is running very rapidly in place and getting nowhere. And when Alice questions her as to why she's doing this, the Red Queen answers something along these lines: because, my dear, in this place, you have to run as fast as you can just to stay where you are. And so the Red Queen effect here in evolutionary terms is that sexual reproduction allows you to produce genetic variation specifically in the immune system and the genes of the immune system, and create enough variety in amongst the genes of the immune system to allow for organisms to keep pace with parasites that are mutating significantly faster than the organisms that they parasitize. Interestingly, we have subsequently discovered that the bdelloid rotifers do have a way of creating or gaining significant genetic diversity, because they, somewhat uniquely amongst eukaryotes, are able to take up DNA sequences from their environment in much the same way as bacteria do, and incorporate these new pieces of DNA into their own genome. So they don't increase genetic diversity rapidly as sexual reproduction might, but they do have ways of introducing new genes and unique genes into their genome by taking up DNA from their environment. So that allows them to maintain a level of genetic diversity that we otherwise might not expect if they were truly reproducing asexually and had no other way of generating genetic diversity. So those are the rotifers. The acanthosephalans are very closely related to one subgroup of rotifers. And in fact, that subgroup of rotifers is more closely related to the acanthosephalans than they are to the other rotifers. So that's led to the idea that these animals may actually be simply very highly modified rotifers. And it's certainly, based on DNA sequence analysis, the acanthosephalans and the rotifers do actually form a single valid clade, a monophyletic clade. So they are now grouped together in this new clade. And again, I've got to look, Syndemata, I believe is the correct pronunciation, Syndemata. So these are commonly referred to as spiny-headed worms. And when you see a picture, and here is a picture, you can see why they're called spiny-headed worms. They use this to anchor themselves to the organism that they are parasitizing, because they are basically gut parasites of vertebrates. They are pseudocoelomate. They have a hemocoel just like the rotifers, no surprise there. They reproduce sexually, and their reproductive life cycle typically involves at least 2 hosts, maybe more. So a pretty complex life cycle. And that's basically all you need to know about these. Then we have the 2 lophophorate clades, the ectoprocs and the brachiopods. Now, the ectoprocs used to be called bryozoans, which translates as moss animals, because when you see them growing, they form a layer of material because they excrete hard material, rocky material, to form an exoskeleton. And then the lophophores extend out of it. So they look a little bit like a marine mossy arrangement. And these are also reef builders, but of non-coralline reefs, meaning reefs that do not include coral polyps. So here you can see an ectoproct called a creeping bryozoan, and you can see the lophophores extended to feed, and you can see the tubes built out of the hard rocky material that they use to reinforce and protect themselves. The other type of lophophorate is called a brachiopod, otherwise known as the lamp shells, and that's because they look a little bit like a bivalve. They have these 2 hard plates that surround the animal. Here you can see the lophophore inside. But what makes them uniquely different from bivalves is that on bivalves, mussels and oysters and the like, the two shells are located dorsal and ventral, with the hinge line being on the ventral side. So, sorry, on the dorsal side, with the ventral side facing out through the opening of the two valves. Those are the two shells that make up a bivalve. They are actually called valves, hence bivalve, two shells. In a brachiopod, the two shells are located on the lateral side, so on the left side and the right side of the animal. So one side here is the left side, the other side is the right side, and the lophophore then extends out from the anterior end of the animal, and dorsal and ventral are located along the slit between the two shells. So it's a slightly different arrangement. So while they may look somewhat like bivalves, they are most definitely not. So those are the two lophophore clades. Then we come to the mollusks. This is the largest of the Lophotrochozoan clade. These are ones that you are almost certainly familiar with because of the snails that you may see crawling across the sidewalk after it's rained. These are also includes the oysters, the clams, the cephalopods, the octopi or octopuses. Apparently, the term octopi is not considered acceptable as a plural. It's octopuses and squids. So the vast majority of these are marine, but there are freshwater mussels that we'll talk about, because there are certainly freshwater mussels that create pearls, just like the marine oysters do. And also because there are snails and slugs that occupy terrestrial habitats as well. So because the body of all of these animals is relatively soft, the vast majority of mollusks have some form of hard shell created out of calcium carbonate and secreted by a feature that is unique to mollusks called the mantle. There are secretory glands along the edge of the mantle that secrete this calcium carbonate material to create the shell. Most mollusks have a single shell, so the typical snail shell is typical of the vast majority of mollusks that have a shell. And in fact, gastropods, the group that snails belong to, are the largest group of mollusks. But the alternative is to have two shells, the bivalve arrangement of oysters and clams. Then when it comes to the cephalopods, the octopuses and the squids, in the case of the octopuses, they have elected or evolved to dispense with the shell completely, which means that their bodies can squash down to a thickness of about 1/4 inch, and they can get through some amazingly narrow gaps. And they're incredibly intelligent along the way as well. Squids have a remnant of the shell, and you should have seen that in the second invertebrate lab. If you watch the video of the dissection of the squid, you will have encountered the remnant of the shell as part of the dissection, and it's internal. A member of the squid family, the cuttlefish, actually retains a larger version of that, but again, it's internal. You can buy cuttlefish skeleton material. It's called a cuttle bone, that you can give to a bird to provide them with sources of calcium. So we utilize those for that reason. The major parts of a mollusk body plan are the foot, the visceral mass, which contains most of the body organs, and then the mantle that surrounds the visceral mass, secretes the shell, and in the case of terrestrial mollusks, provides a modified gill apparatus for gas exchange. So here you can see the major features of a typical mollusk, in this case a gastropod, a snail. So here's the foot used for crawling or digging. and in the case of some bivalves can also be used for swimming by wriggling it around. You can see that it's an open circulatory system. So you have a hemocoel, not a coelom, you have a hemocoel. And the fluid called hemolymph diffuses through the tissues of the organs and provides nutrient and oxygen obtained by oxygenation through the gill. The visceral mass also contains the highly folded digestive tract, and it is a true digestive tract or a true gut. It has a mouth and an anus with the cylinder connecting them together. The nerve system consists of a ring of nerves around the anterior end of the gut and a pair of nerve cords that run through the foot to coordinate the rhythmic contractions of the muscles of the foot to allow the animal to crawl. In the mouth is a hardened structure called the radula. It's a series of teeth that are hardened. In herbivorous mollusks, this is used to scrape algae off the rocks to use as food. In predatory snails, and there are predatory snails, this can be modified as a tool to drill a hole down through the shell of other mollusks for the purpose of eating that mollusks. And I in another life, when I was much younger, used to collect shells, mollusk shells, and I have a number of shells that have a nice, perfect little hole drilled through it, which is evidence of a snail called a moon snail that actually feeds in this manner. It drills a hole down through the shell of another mollusk and then inverts its feeding apparatus through that hole to scrape out the soft body of the mollusk. Then in a small group of snails called cone snails, and these are referenced in the textbook for the first time that I'm aware of. I've not seen them referenced in the textbook before. The radula or the teeth of the radula are actually modified into little harpoons that are used to disable fish, because these are snails that eat fish. And they have very potent venoms, just like the cnidarians, in order to be able to paralyze the fish in order to be able to eat them. And in the case of at least 3 cone snails, the geographic cone, the golden cone, and the textile cone, the venom of those cone shells is sufficiently potent enough to kill a human. And they have, in the case of the geographic cone and textile cone, been recorded to have killed humans. And the golden cone is suspected of killing humans. Now, I do have shells of textile and the textile cone and geographic cone in my collection, but I did not collect them myself. I actually bought those. I don't know too many people who would legitimately want to collect those by hand, because one of the features of these cone snails is that they can extend the proboscis that contains the radular apparatus sufficiently out of the shell to reach around and stab you while you're holding the back end of the shell. And there is no known antidote to these venoms. So if you get stuck, you are going to die. And it is very, and as I said, in the case of these 3 snails, it is incredibly potent. But that's because they're preying on fish. So let's look at the 4 classes of mollusks that represent the largest classes. So the first is class Polyplacophora, meaning many plates. And this is a reference to the fact that the shell is broken up into 8 plates that cover the dorsal side of the animal, linked together by cartilage so that it is flexible. These animals live in the tidal zone, so they are regularly exposed to dry air when the tide is out and then underwater when the tide is in. They feed when the tide is in by creeping over the rocks and scraping algae off the rocks with the radula. When the tide is out, they cling to the rock so tightly that the shells form a seal with the rock surface and prevent them from dehydrating. They also have very primitive light detecting organs that allow them to detect the presence of shadows, and immediately a shadow falls on them, they will suck down onto the rock, and it is extremely difficult to get them back off again. They can cling very tightly. So here you can see a typical chitin. Just as an aside, one of the other books that Darwin published, because he did publish other manuscripts other than On the Origin of Species, was a description of the chitins and that inhabit the seashores of Great Britain. It's not something that many people know about Darwin. Now you do. The next group and the largest group are the gastropods. This translates as head foot. And so the muscular foot is used for crawling. Sorry, stomach foot, not head foot, stomach foot. Cephalopod is head foot. Gastropod is stomach foot. So these are predominantly marine. So these are snails and slugs that live in marine environments or freshwater environments, but of course there are terrestrial representatives as well. A slug is simply a gastropod that lacks a shell or has a very small internal shell. Oops. So here's a classic terrestrial snail with its shell, and here is a sea slug, otherwise known as a nudibranch. Now, interestingly, some nudibranchs eat coral polyps, and you can see that this nudibranch is actually cruising across the surface of a coral formation. You can see the little polyps in here. So these eat coral polyps, and they take advantage of the fact that the coral polyps are cnidarians with those cnidocytes. And rather than digest the cnidocytes, they absorb the cnidocytes and they translocate them to these extensions on the back of the slug. I'm not entirely sure what we call them. They look a little bit like tentacles. I'm not sure what the correct name for these is. But these are covered in, or these contain cnidocytes that are functional, just as they were functional in the cnidarians from the coral polyps from which they were extracted. And this acts as a way of protecting the nudibranch, the sea slug. So let me put here, AKA. Nudibranch. And yes, nudie here does refer to naked, because it doesn't have a shell. It's a naked body. That's where the name comes from. So these cnidocytes serve to protect the nudibranch. That's why they're brightly colored, as a warning to a predator that might want to eat the nudibranch, that if they do, they will be in a world of hurt because of the active cnidocytes that are present. Nature always warns of the presence of something that's dangerous. Bright colors, red, for instance, or the bright colors of the poison arrow frogs. Those are the way nature warns. Handle this at your own peril. Touch this at your own peril. Eat this at your own peril. And you can't say that you weren't warned. So one of the interesting things, and I was somewhat puzzled that the book doesn't mention this, is that the body of a gastropod undergoes a change called torsion. It means that the relationship between the dorsal and ventral organs in the body undergo a rotation of 180 degrees so that they are in a new orientation to each other. And so I've included a diagram from a previous edition of the textbook to show this. So normally we expect the mouth to be higher than the anus to prevent the ingestion of waste that might be in the environment. But because of the effect of torsion, it rotates the organs within the visceral mass and relocates the anus above the level of the mouth. And this is unique to gastropods. So only gastropods undergo this torsional rotation. So this lays out the body organs of a bivalve. Well, we're changing to bivalve, yeah. So bivalves are the clams, oysters, mussels, and scallops. So these are the mollusks that have two shells that enclose the body of the animal. Remember again, these are located on the lateral side of the animal and are hinged along the dorsal line with the ventral side of the animal facing outwards. In the lamp shells, the brachiopods, the shells are located on the dorsal and ventral side and are hinged on the lateral line. Just want to state that because I'm not sure whether I said that correctly when I was talking about them. A little bit fuzzy, and I just am afraid that I may have said that incorrectly when we were talking about the brachiopods. Anyway, so the shells are held together, or the valves are held together by a pair of muscles called adductor muscles. These are very, very strong muscles. If you've ever had to try and shuck an oyster, you know just how tough these muscles are to extend. They are very, very strong muscles. And yet, the major predator of clams, oysters, and mussels are sea stars. And when we talk about sea stars, echinoderms, I'll explain how they manage to get around the adductor muscles of mollusks. It's quite a clever strategy. So when we take a look at the body cavity of the internal features of the bivalves, you have a water current that is generated by two openings called siphons, the in current where the water comes in, the X current where the water goes out. The water passes across the gills to oxygenate the hemolymph, because again, it is an open circulatory system. And so we don't talk about blood, we talk about hemolymph. The food gets filtered out as through the gill apparatus and passed into the mouth of the digestive tract. The food then gets processed in the gut, absorbed through the intestine, and secreted out the anus, which secretes into the X current to carry the waste back out of the body of the organism again. You see the mantle that lines the inside of the cavity. It secretes the shells. It can also extend out and cover over the shell, and in many cases, it can allow the mollusk to blend into the background and hide itself in the environment. This is true both of the gastropods and also of the bivalves. Trying to think what else I need to tell you at this point. There are a couple of other things that are coming up, but I'll tell you about that as we address them. I thought I had more information in there. Maybe I've taken some of it out as a consequence of this getting really long. There are two sexes. There are males and female clams, but it is essentially impossible to tell the difference. But they do release their eggs and sperm into the water column, and the fertilization takes place in the water column. So it's external. Then we have the cephalopods. These are the head-foot animals. So what looks like the head of an octopus or the head of a squid is actually a modified version of the foot. And then the mouth is also different from the mouth of the other mollusks in that it has a hard chitinous-like material that forms some beaks so that they can tear material apart. And these beaks can actually be quite sharp and quite vicious when they lay into the prey that they are trying to tear apart. And that can include humans. One of the things you never want to do is mess with an octopus or a squid, because if they wrap their tentacles around you and start to bite you, they can literally rip big chunks of flesh out of you, because those beaks are very hard and very useful. Cephalopods are unique amongst the mollusks in that they do have a closed circulatory system. So here we can actually talk about blood, because remember, blood is the term that we reserve for closed circulatory systems. If it's an open circulatory system, then it's hemolymph. Cephalopods are also extremely intelligent. They are good at problem solving. They are good at working through mazes. They can also learn and create memories. Check out YouTube for some videos of octopuses solving mazes. It is amazing to watch them work. And you also need to remember that their eyes are functionally equivalent to ours. They have a flexible lens with muscles to change the curvature of the lens to form images. They're armed with a very, very intelligent brain. We think that they are at least as intelligent as our dogs, maybe even more intelligent than that. They certainly have the ability to solve problems that some primates find really difficult to solve. I remember watching a video of a crab-eating monkey going up against an octopus for extracting a crab out of a container. Now, the container had an opening in it that was not large enough to allow the monkey to put its hand into the container, grab the crab and pull it back out of the opening again. It could stick its hand in, but as soon as it grabbed the crab, the fist was too large to come back out of the opening again. And as much as the monkey tried, it could never get the crab out, because it consistently tried to grab the crab and pull it out. And it didn't come up with an alternative solution. It could not see another solution to the problem. If you watch an octopus faced with the same problem, it doesn't immediately try and grab the crab. It looks over the situation, and you can almost imagine it sizing up the problem and trying to decide what is the best approach. And then within a minute or two, the octopus clearly comes up with the solution. It crawls in the opening of the container into the container and consumes the crab inside the container. It doesn't even bother to take the crab out. It gets into the container with the crab. Yeah, they're really intelligent. So here are members that you may be familiar with, the squid and the octopus. You may not have encountered a chambered nautilus, but you may have come across the shell. The reason why you don't see chambered nautiluses is because they live deep in the ocean. These are usually below 1,000 feet in the ocean. And they have multiple tentacles, and they are the only cephalopod to retain a shell. And their shell is very similar to the ancestor of the mollusks, the ammonites. That are, when we look at the evolutionary history of mollusks, we look at ammonites as the ancestors of the mollusks, and the chambered nautilus shell looks very much like an ammonite shell. So here we have the mention of them here. So the ammonites were very commonly found in the oceans all the way through to the end Cretaceous extinction. So they disappeared with the dinosaurs. The same extinction that took out the dinosaurs also took out the ammonites. In fact, the extinction, the end Cretaceous extinction in marine environments is much more dramatic than in terrestrial environments, because marine environments are far more sensitive to changes of temperature and changes of oxygen content than terrestrial environments. So a small change in temperature can be catastrophic in a marine environment. And not surprisingly, a large number of species, we think over 90% of all marine species at the time, went extinct. The net number of species that went extinct, terrestrial and marine, was somewhere more in the range of 70%. But in the marine environment, we think upwards of 90% of all species went extinct, simply because of the sensitivity of marine environments. So those are the mollusks. The final Lopetrochozoan is the annelids. Now, we used to split the annelids up into 3 classes, but subsequently, we now believe, on the basis of DNA sequence analysis, that those 3 classes really belong as a single clade, or perhaps 2 clades. The book references it as 2 clades, so we will take that approach. That seems to be consistent with what my sources are saying as well, that it is more appropriate to consider it as 2 clades. So let's take a look at the annelids. So here we have a worm that is best described as a segmented worm. That is typically the common name that's given to them. They have a true body cavity, so no hemocoel or pseudocoelom. They have a true coelom. The body is divided up into a sequence of segments, and the organs in each segment are repeated from segment to segment, although there are in some of the anterior segments specialized organs, and also in the middle region of many annelids, there are again some specialized organs for reproductive purposes located in a region called the clitellum. But basically, the organs that show up in one segment are repeated in all of the segments down the body of the worm. So the 2 clades that are currently best supported are the Urantians, otherwise known as mobile annelids. Now, this can imply crawling, swimming, even burrowing, but burrowing in this circumstance is far more rapid than the burrowing that we see in the second group, called the sedentarians, otherwise known as the sedentary worms or the slow burrowing annelids. So the Urantia include most of the individuals that were in the phylum Polychaeta, and Polychaeta were characterized by being marine worms and having on each segment a pair of appendages called parapodia that have multiple little hairs on them called setae, hence the name polychaete, many hairs. But also these parapodia are highly vascularized, and so they are used for gas exchange as well, and can also serve as a means of locomotive. They are parapodia, parallel legs, so they can be used for locomotion as well for those that crawl across the surface. Some drift as plankton, so these are the swimmers. Vast majority of them crawl, but a few of them do live in tubes that they create themselves by taking sand and shells and gluing them together with mucus to form a tube-like arrangement around the outside of them. They are either predators or grazers to a large degree, but those that live in tubes are often filter feeders. So here you can see a classic Urantia. You can see the parapodia with the little hairs on it. And the other feature about the Urantia is that there's always a fairly clear anterior to posterior end, something that is not quite so clear-cut about this sedentaria. So when we look at the sedentarians, these are typically less mobile. These are the true tube worms. which anchor themselves to the substratum and spend their lives in tubes that are built out of a hard material, typically a calcium carbonate material that they secrete to build the tubes, and they remain anchored to their substratum and don't move out of it, and they are strictly filter feeders. Alternatively, they can be burrowers. Those would be your classic earthworm. This also includes the leeches as this, amongst the sedentarians. The vast majority of leeches are either freshwater or terrestrial. Yes, we do associate leeches with feeding on blood, but that is only a small number of the total leech population. Most of them are carnivorous and feed on small invertebrates that they can capture in the environment. Those that are blood feeding typically have a jaw-like apparatus that allows them to slice through the skin. Many of them have blades that work like the blades on a pair of scissors that slice through the skin. They can also secrete an anesthetic that numbs it so that you don't notice that that's what they're doing. And they used to belong to a clade called Hirudinea because of the fact that they secrete a protein anticoagulant called hirudin that allows the blood to keep flowing so that they can ingest their blood, the blood that they are consuming for their meal. And the average leech that consumes blood will take up about a 10-fold volume of blood and may not need to feed again for several months. Now, in the past, leeches had a medicinal use when it was popular to bleed people because it was considered that bad blood was the cause of disease. And so if you wanted to cure someone of their disease, you bled them. And before they created specialized knives and all sorts of tools for bleeding patients, many quote unquote doctors would use leeches to bleed their patients. Of course, once it was the germ theory of life took hold and it was understood that many diseases were caused by infections, the whole notion of bleeding people fell by the wayside, and so did the use of medicinal leeches. But interestingly, over about the last 25 years or so, medicinal leeches have made a comeback because of their ability to, number one, remove blood, and number two, secrete the hirudin to prevent the blood from clotting. So where might you use this? Well, if you have a buildup of fluid in an appendage, particularly blood, a leech might be applied to remove the fluid to allow for relief of the pressure that the fluid is creating. Or if you've had an amputated appendage, a finger or something like that reattached, your healthcare team may attach one or more leeches to the reattached digit to encourage blood flow, because one of the primary reasons why a reattached digit may fail to survive is because of lack of blood flow. But a leech's ability to, number one, stop the blood from clotting, and number two, promoting blood flow as a consequence, increases the chance of the digit actually reattaching and all the tissues and blood vessels and everything connecting back up again, because removing the blood and preventing the blood from clotting stimulates the growth of new blood vessels. And so medicinal leeches are now actually being actively grown by specialized companies for the purpose of using in modern medicine. So here you can see a leech attempting to attach to this thumb, but the individual is wearing a plastic glove to prevent that. The leech is nonetheless still trying to do so. And you can see how segmented the body is, a classic annelid segmented body. Then you have earthworms, also included amongst the sedentarians, because they slowly burrow their way through the soil, ingesting the soil, extracting organic material out of it, metabolizing it, and then secreting their waste called casts back into the soil. And that actually is beneficial for the soil. Charles Darwin, with his son, Leonard, actually carried out an experiment. And if you visit Darwin's house outside of the village of Down in Kent, in southern England, you can actually see the experimental apparatus in the lawn. I've actually seen it. It's a concrete disc that rides on a metal shaft that they sank into the ground. And they measured the rise and fall of that concrete disc as a way of measuring the activity of earthworms churning up the soil underneath. And Darwin then extrapolated from the data that they collected to figure out just how important earthworms were to agriculture in Great Britain. And he estimated that they returned about, I think it's about 2 million pounds of metabolized earthworm poo back into the soil. Pretty important source of easily accessible organic material for the plants that are growing in the soil. Now we do know that while earthworms and annelids in general are hermaphrodites, they do exchange sperm. So the way they do this, and I'll see if I can illustrate this with a nice diagram. Here is one earthworm. And this is the clitellum. So we label that ant and post. And one of the things about the sedentarians, and particularly earthworms, and what used to be called oligochaeta, is that the anterior and the posterior end are sometimes very confusing to identify. So the two worms will line up next to each other and will slide past each other. So this worm's going this way, this worm's going this way. The clitellum secretes a foamy mass that surrounds the body of the worm that they crawl through. And they exchange sperm by depositing a packet of sperm into the foam mass of its partner worm. So they exchange sperm that way. The sperm get deposited into this foamy mass. The worms then crawl away, crawl out of the foamy mass. It seals over at each end. Within that within the foam are the eggs that have been deposited by the worm, and now the sperm from the mating partner. Fertilization takes place in this moist foam environment. The larvae develop in there, and eventually the little juvenile worms emerge out of this little foam packet, the outside of it being hardened to protect the inside from dehydration, while the worms move very quickly from zygote to larva to little juvenile worm. Excuse me. So yes, while they may be hermaphrodites, they do actually exchange sperm. They don't self-fertilize. So here you can see the body plan of a typical earthworm. So within each segment, there is a part of the dorsal and ventral blood vessels that extend the full length of the body of the worm. In each segment, there is a ring vessel that connects the two longitudinal vessels together. At the very anterior end of the worm, there are 5 rings that function as hearts to pump the blood down the body of the animal. Within the digestive tract is a complete digestive tract, mouth, anus, cylinder to connect them together. You have a pharynx where the food material is ground up, an esophagus where it is further ground, a crop where it is ground to its final product and passed into the gizzard, and then on into the intestine for absorption. The waste is then excreted from the anus at the tip of the worm. Each segment externally also has a pair of these little hairs called setae or setae. These grip the soil as the worm burrows through the soil. Within each segment, there are two layers of muscle, the longitudinal muscle on the outside of the cavity and the circular muscle that surrounds the digestive tract. It is the rhythmic contraction of the circular muscle and the longitudinal muscle that sends the ripples down the body of the worm. And so each segment rhythmically shortens and then lengthens, and that rhythmic contraction in conjunction with the gripping of the soil by the setae then drive the body of the worm through the soil. Then within each segment, there are branches of nerve cords that organize the contractions of the muscles coming off the ventral nerve cord, again, a ventral nerve cord running down the body of the animal. And each segment has a pair of nerves that come out to drive the muscles of the segment. Each segment has a nephrostome, which is a secretory organ, I guess, for waste disposal, and a metanephridium, also for waste disposal. How big can an earthworm get? Well, the average earthworm is usually somewhere between 3 inches and 6 inches in length, depending on species, but there is a giant earthworm that lives in a region of northern Victoria and southern New South Wales called the Gippsland region that can get over 6 feet in length. These are threatened with extinction, however, because of agricultural practices in the area. It's also a citrus growing area, and because of the irrigation practice, it's creating saline soil that is putting the earthworms at risk. So this is a species of earthworm that is currently threatened with extinction as a consequence. So those are the Lophotrochozoans. Quite a lengthy presentation, so we are going to take a break. So here is the concept question for the break. Earthworms are often described as a tube within a tube. Explain what is meant by this description. I'll see you on the other side of the break with the answer.